Since the introduction of genetically modified foods, controversy surrounding the topic has become increasingly apparent in the media owing to its health and ethical flaws.
Today, we'll be delving into the controversies, science, and advantages of genetically modified food. We'll be deriving our content from Harvard University's Gabriel Rangel in his article titled Corgis to Corn, a brief look at the long history of GMO technology. This article was published on August 9, 2015.
Genetically modified foods are food items produced from organisms, including both animals and plants, that have had their DNA manipulated in some way to obtain a chosen and typically beneficial trait. Genetic modification differs from traditional crossbreeding of organisms as results are obtained through genetic engineering rather than breeding. Although modern genetic modification was discovered back in the early 1900s, genetic manipulation of food dates as far back as 10,000 BC, involving artificial selection. Current means of genetic modifications are derived from these precursors of modification, and after the discovery of DNA in the 1900s, genetic techniques have made it possible to directly alter the DNA and genes within food to suit the agricultural and dietary needs of today. The characteristics of every living organism is determined by its genetic makeup and interaction with its surroundings throughout its lifetime. The genetic makeup of an organism is called its genome, which consists of its DNA. The genes hidden within the DNA carry instructions for proteins, hence determining the organism's qualities. Today, the techniques of genetic modification of food employ modern ways of identifying these desirable traits from the genome of a certain organism that may result in similar benefits if implemented into another organism. Then, the selected genes that code for these specific qualities are transferred between the chosen animals or plants. After the desired trait of the source organism is identified and extracted, the process of genetic modification of food begins. The first stage of modification is the transferal of a gene from one organism to another organism. This can be achieved using numerous techniques, two of the most commonly practiced, the particle bombardment method and the agrobacterium tumefaciens method. The particle bombardment method utilizes a particle bombardment device or gene gun to penetrate a cell wall so that the genetic material of interest can be transferred into a cell. Small particles of metal, usually tungsten or gold, are coated in plasmid DNA. These metal particles are accelerated with air pressure and bombarded into the plant. As the genes are released from the metal particles, they fuse with the DNA within them. The more common method is the agrobacterium tumefaciens method. Agrobacterium tumefaciens is a bacterium that, like many others, transfer their DNA into their host cell as part of their normal life cycle. Utilizing this property, the gene of interest is transferred into this bacterium, which then transfers the chosen gene into the genome of the target plant or animal. If the target organism is a plant, the cells that have been genetically affected by the gene of interest can be grown into a new plant. If the source organism or the organism that the gene of interest was derived from was the same species as the target, the affected organism is referred to as cisgenic, whereas if the source is from a different species, the organism is called transgenic.
Here is a video showing how a gene can be inserted into DNA.
Double helix. Scanning in the case of EcoR1 for the base letter sequence GAATTC. The enzyme then cuts the plasmid at this specific point, allowing a new piece of DNA to be inserted.
Genetic modification of foods was an immensely important discovery for both scientists and the food industry. Genetically modified foods can have notable benefits over naturally occurring foods. The modifications can range from resistance to pests, increased yield, increased nutrients, improved health benefits, and being longer lasting. A famous example of this is the Flavor Saver tomato, the first licensed genetically modified food. It was created to counter the issue of its short ripeness time. Genes were inserted to keep its shape, firmness, and flavor for a longer period of time. Scientists are still working on the development of genetic modification of food, and it is likely that near all of our food may be genetically modified in the future.
The introduction of genetically modified food has caused a big change in the agricultural industry that affects both producers and consumers. GM foods have positively affected the agricultural industry in developing nations. Many developing nations, especially in Africa and Asia, are experiencing a high demand for food to feed their country's growing populations. Genetically modified foods help meet this demand as more produce and higher quality products are made available to the public through the altering of genetic material in plants and livestock. Not only does GM food aid developing nations, but it also helps all food shoppers and eaters in most of the world's countries. Genetic modification keeps the shelves at the supermarket stacked by reducing the risk of crop failure and diseases. However, for non-GM farmers, GM foods pose a disadvantage for their businesses, as GM food companies hold a competitive advantage in terms of farming efficiency. Whilst genetically modified food has helped work towards solutions in major global issues, such as a high food demand in growing populations and a lack of agricultural resources, there are still some issues and concerns surrounding the topic. One of the most recurring objections to GM foods is the potential health hazards that they pose to the public. In September 2012, the Seralini study was published in the Food and Chemical Toxicology Journal. The study researched the effect of feeding rats genetically modified corn compared to naturally grown corn. The results of the study found that the rats that were fed the genetically modified corn experienced a higher mortality rate as well as an increase in body tumours. Unsurprisingly, this study raised a lot of eyebrows and caused many concerns about the safety of GM food. Even though the results of the study have since been proved inconclusive, the safety of GM food has remained a common concern amongst the public. Another issue that GM food poses is the potential outcrossing of modified genes into wild populations. Outcrossing refers to the risk of GM crops mixing with natural or conventional crops. The introduction of GM crops into wild populations could potentially cause a snowball effect, resulting in large-scale changes and damages to the ecosystem. Due to some of these issues mentioned, many of the world's countries have imposed restrictions or bans on GM foods. These countries are shown on the map presented on the screen. How will the technology used in GM foods improve or progress in the future? How are the genes necessary for genetic modification extracted from the source organism? Is it possible for humans to live solely off GM foods?